Oh no, that was brilliant. Okay, as Mitch said, I'm from the English department. I teach mostly freshman composition. Many of you may remember it from a number of years ago. And I also, when I get an opportunity, teach a course in Tolkien and another course in science fiction. And I think that's probably why they picked me, because I at least have that much. And one of the reasons I came is that I'm stealing ideas. So what you hear today may be transformed into a novel that comes out in about 5 years. But in any case, you were here when it happened. So anyway, given my background in literature, I'm really fascinated with what these people are presenting. We're going to have all three people go, and we're going to hold our questions and comments and responses and postulations till after all three have gone, and then we can have a more general forum. First, I'd like to present Laura Berner, who is doing work on, what is it, imaging, brain neuroimaging in relation to Bulimia and Nervosa. Actually, one of the great machines that she uses reminds me of the ancient Nordic dragon Fafnir, and so I'm curious how that's going to come out. So this is Laura Berner.
Thank you for that introduction. I wanted to start with lots of thank yous. So first, thank you to the GSA for having me here to speak today and inviting me. I also want to acknowledge contributors on this project, including my dissertation committee, my mentor, Michael Lowe, and a special shout out to Andrew Caspery, who designed some of the software that I'm going to be telling you about today. Also, just to briefly acknowledge my funding sources. So, to start with, just a little bit about me. I started doing research in my sophomore year as an undergrad, doing animal models of eating disorders research. So I spent a lot of time getting bitten and scratched and basically looking into neurotransmitters that could tell us more about what goes into the development and maintenance of binge eating behaviors and how that could help us inform future treatments. Then I worked as a research assistant, getting experience delivering test meals and looking at treatment outcome in folks with anorexia nervosa and bulimia nervosa, as well as binge eating disorder, and got some experience there doing neuroimaging research using fMRI in folks with bulimia nervosa. Which brings me to here at Drexel, where almost all of my research focuses on integrating biology, which is sort of my undergraduate roots, with more psychology, to really build a full and complete understanding of eating disorders. And ultimately the goal is to use that information to help us better inform both prevention efforts and treatment efforts. So in terms of what I'm going to be talking about today, I'll tell you a little bit about what bulimia nervosa is, because not everyone here might know. I'll tell you a little bit about some previous work that I've done as a graduate student here, but mostly be focusing on what my current research is, which is very much in progress at the moment. So bulimia nervosa is an eating disorder that's characterized by episodes of binge eating, and that's defined currently by the field as eating a large amount of food in a discrete period of time, and importantly, experiencing a sense of loss of control over your eating. So people describe that as sort of feeling like it's hard to stop once they've gotten started, or feeling like driven or compelled to keep eating. It's also characterized by compensatory behavior, so things like self-induced vomiting, taking laxatives or diuretics to sort of undo the effects of eating to promote weight loss or influence shape. The disorder is associated with significant medical complications and high rates of comorbid psychopathology. It affects 1 to 3% of women, and the best treatments that we have still aren't as good as we'd like them to be. About 30 to 50% of folks don't respond to the best treatments that we have, and about 40% of them remit, so they go back to having bulimia nervosa after they've gotten better. So there's this clear need for a better and more complete biopsychosocial understanding of the disorder to inform prevention, treatment development, treatment selection, and improving the prediction of outcomes. So who's going to do well with what treatment? So some of my research has focused on sort of biological variables and what that tells us about symptoms. So looking at a variable that my mentor has really been instrumental in developing, which is weight suppression, or the difference between somebody's highest adult lifetime weight and their current weight. So how far is their current weight below that? And it turns out it's a really predictive and robust variable in these folks. One study we did actually showed that women who were more weight suppressed at entry to a study took longer to recover from bulimia nervosa 8 years later. So there may be some suggestion there that these folks are stuck in sort of having the psychological influences of wanting to maintain a lower weight, but also biological influences of their body kind of wanting to be at a higher weight, so they're constantly fighting that. And weight suppression might be a good way to kind of capture that integration of biology and psychological pressures. Other research that I've done has looked at sort of patients' perception of loss of control eating episodes and how much their perception of the size of that episode matters. And in looking at some folks who are receiving treatment for bulimia nervosa, we found that people who were viewing episodes that were actually sort of normal sized, so eating kind of a normal meal, but it felt large to them, and they felt a sense of loss of control. The more episodes like that that a person had at admission to treatment, the more severe their sort of general psychopathology and eating disorder symptoms were. And folks that were having episodes that they didn't think were large, but that they experienced a sense of loss of control during, those folks actually do better in treatment. And the frequency of those episodes go up over the course of inpatient treatment. So now to the good stuff, sort of what I'm working on now, really focuses on inhibitory control deficits in bulimia nervosa. So individuals with bulimia demonstrate marked general impairments in self-regulation. A lot of studies have shown that this population has higher rates of gambling problems, self-injurious behavior issues, as well as substance use disorders. And neuropsychological testing that assesses self-regulation in bulimia has yielded somewhat mixed results. So that's kind of confusing in light of what we're seeing clinically. A lot of neuropsych tests that use kind of button pressing responses, some have found that folks with BN perform worse than healthy controls who've never had an eating disorder, and some studies find that people actually perform just about the same, whether they have bulimia or they're a healthy control. I'm interested in understanding how the brain fits into this whole picture. So in terms of what the brain does to help us exert an inhibitory control response, there's a circuit that links your prefrontal cortex, and I'll try to keep this jargon-free, so the front of your brain to more the center of your brain, or what we call the striatum, and that subserves the ability to inhibit a response. When you sort of see a cookie there on the table, that's the circuit that's activated that keeps you from reaching for that cookie. So there seems to be this important role, specifically for the inferior and middle frontal gyrus, which is right at the front of your brain, that seems to be kind of the central executor of self-regulatory control. And importantly, go-no-go tasks, which I'll tell you a little bit more about in a second, really activate this region pretty reliably. Some studies have also shown that efforts to suppress thoughts or urges to salient cues like food, drug pictures, or even thoughts about smoking in folks who smoke, efforts to suppress those thoughts also activate these similar prefrontal regions. So in terms of people with bulimia, what do we know about their brains? There haven't been that many studies done that sort of look at the brains of individuals with bulimia. But those that have been done suggest that there is deficient prefrontal cortical activity compared to healthy control. So that part of their brain is not activating as much for some reason as healthy folks. And this may be key to that sense of loss of control that I told you about in the beginning that characterizes binge eating episodes. That sense of it's hard for me to stop once I've gotten started. So I wanted to really move this into more direct applications to understanding eating disorder symptoms. So I created a novel task that involves eating. So no study has utilized a task so far in which the response is eating related. There have been some neuropsych tasks that have been developed where the stimuli are changed from neutral stimuli to food stimuli, but the actual response has nothing to do with eating or food. And so a go-no-go eating or sipping paradigm could enhance our understanding of the ways in which brain activation more directly relates to eating disorder-specific behavior. It's also a liquid consumption task, right? So from the start of the task to the end of the task, you're becoming more full or more sated, which sort of mimics what we see in real life. Over the course of a binge episode, somebody isn't staying the same degree of hungry or full, and so we want to mimic that piece as well. So this is an image, I don't know if you can all see it, that's captured from one of our participants completing this sipping task, and you can kind of see the basic setup of what that looks like, and I'll explain more in a bit about it. So the other piece of this is that we want to understand what's happening in the brain while folks are trying to exert inhibitory control over an eating response. And a problem is that we can't really do that so well using fMRI, because if any of you have ever been in an fMRI scanner, you know that you have to stay super, super still. And if the head moves really beyond a millimeter, you lose a lot of the data. If you're asking somebody, especially somebody with an eating disorder, to perform an eating task that involves repeated sipping and swallowing while they're in an fMRI scanner, your data would be pretty messy, unless you were very specifically telling them what to do exactly when. So we wanted to use a neuroimaging method that would allow for some more motion and that would allow for someone to be sitting up, which you can't do with fMRI, and eating in a more realistic way. So we are using functional near-infrared spectroscopy, which is a mouthful, or fNIR for short, and this particular model of it was developed by folks at Drexel. And the device is basically a headband, sort of as depicted in that picture in the top left corner, that covers those regions that I was telling you about that are really thought to be the central executors of self-regulatory control functions. There have been found to be significant correlations between fNIR and fMRI data, and fNIR is really ideal for more ecologically valid study of a behavior that's symptom-related in somebody who's actively ill. So we're already asking people to do something that's going to be kind of upsetting. And so we want them to be as comfortable as possible and to not have an intrusive clanging magnet going on in the background, which would be the case with fMRI. So the goal of this study, my dissertation, is to compare fNIR measured brain activity during a standard button pressing go-no-go task to those of women with BN, as well as to compare how well they do on the task, and then to look at how their performance on the task correlates with their brain activation patterns. And then using the sipping task, we're also looking at comparing brain activation between healthy women and women with Bulimia or Nervosa, and comparing how well they do on the task, and that's where the cool new software comes in, and demonstrating that activation, that go-no-go sipping task-related changes in the front of your brain correlate with behavioral performance on the task, and also to look at how performance on the task and activation during the task relate to how frequently someone tells us they've been binge eating in the last 3 months. So we're aiming for a sample of 35 women with Bulimia Nervosa and 35 healthy controls. At this point, we've run on the up-to-date new paradigm 5 healthy controls and 1 woman with BN. We hope to match the groups for age, BMI, and if possible, total IQ score. Otherwise, we'll have to control for them later. So folks come in and they fill out a bunch of self-report questionnaires. So we'll be able to also look at the ways that sort of the performance data and the brain data relate to things like how much somebody reports having difficulty controlling their eating, how much somebody reports having difficulty controlling any of their behavioral responses, as well as their weight and their weight history, and how hungry and full they report being at the beginning and the end of the task. So first, they complete a standard go-no-go button pressing task. This is a really kind of standard measure in the field for assessing inhibitory control. You see a bunch of slides in order that display letters, and you're instructed to press in response to any letter except an X. So they're supposed to withhold the response when they see an X, which is only 15% of the targets that they're seeing. So you build up this prepotent response tendency, so you're pressing, and then you have to stop, and that requires inhibitory control. In the go-no-go sipping task, the instructions are exactly the same. Sip and swallow once instead of button press when you see any letter except an X. So the instruction is sort of maintain people have practice doing it by that point. And we place 1,500 grams of a yogurt shake in front of participants, and an HD camera that records liquid movement in the straw while the participant is completing the task. And that allows for us to code whether somebody responded or didn't respond to the stimulus that they saw. So in pilot testing of this paradigm with shorter versions of each of the go-no-go tasks, we did find the expected patterns of activation. These are all from healthy controls who completed our pilot task. In the first panel, the difference between the first and the middle panel are just how stringent we were with our significance criteria. But if you look sort of at the middle panel, which is a little bit more lenient in terms of our p-value, you can see that those same prefrontal regions that I was telling you about that we would expect to be more activated during no-go trials were more activated when people were trying to inhibit their response on the standard button pressing task. And on the third panel on the far right, that's activation that's associated with the sipping task. You can see that the signal is definitely weaker, but it's activating the exact same regions that we would expect to be activated. So sort of proving the feasibility of doing this. The latest sort of development is this video analysis software that we now have, where we can actually upload these HD videos of people sipping, set sort of reader windows on the straw to record when liquid is moving up through the straw, and it spits out sort of this timestamp of when that happened, which we then can map up with the timestamps of when the people saw each of the stimuli. So it allows us to code whether someone responded correctly or incorrectly to each stimulus. So unfortunately, I can't present to you the data that we have from the people we've run so far, because I'm going to be presenting it hopefully somewhere else. But I will tell you that we've run one woman who has Bulimia nervosa, and she reported feeling like she did binge eat during the task, and felt a very strong urge to purge after the task, and had a lot of difficulty both on the button pressing task and the sipping task. Her performance looked worse than healthy controls. That being said, it's an N of 1, so we always have to interpret that with a lot of caution, but looks promising so far. So we are expecting that women with BN are going to show that pattern of less activation on the no-goes in the fronts of their brains, and that they may exhibit deficient performance as well on both of the tasks. If our aims aren't supported, we're still going to be getting behavioral data that's really important to the field and helping us better understand and standardize a way of looking at loss of control over eating in a laboratory-based eating task. So this is really the first investigation of neural activity during an eating self-regulatory control task in women with bulimia and in healthy controls. That's it. Thank you.
Thank you very much, Laura. And I ask you again to hold your questions until after the other two people finish. Just now, I will present Patrick McLaughlin, who has gone out of doors, not working in the lab, but he actually gets his hands on frogs and swabs them. And so this is going to be a real, I don't know if this is going to be an actual hands-on sort of a presentation, but
they're in the back.
Yep, in the back. Okay. Wow. There's the food is gone. Wow.